Kalibiome postbiotics are a new call of active specialties that represent the future of cosmetics industry. Today, the maintenance and strengthening of skin microbiota is the key to guarantee the wellness and a good functionality of the skin. Kalibiome is introducing a new class of specialties for dermatological applications that represent the most advanced frontier of microbiota care. This innovation was developed by the laboratories of the Humanitas University of Milan, with a scientific team worldwide recognized as one of the most relevant players in the knowledge of microbiology and microbiota. Now, let's see in detail what this innovation consists of. In compositional terms, Kalibiome postbiotics are oligopeptides, fatty acids, and biosurfactants released by Lactobacillus paracasei through a controlled biofermentation, enabling to obtain the above substances in consistent amounts and with a unique purity yield on a reproducible basis. In functional terms, these postbiotics are the messengers and the actual effectors of the bacteria beneficial actions on the skin. Contrarily to probiotics and similar actives, Kalibiome postbiotics show a unique safety profile because of the total lack of bacterial fragments, bacterial wall, and toxins in their nature. This optimized safety profile is demonstrated in a scientific publication of a medical journal, Gut, through a study carried out on the main headquarters of the bacteria in the human body, the gut indeed. The study proves that individuals affected by the Crohn's disease, a pathology characterized by prolonged events of local acute inflammation, when treated with commensal bacteria of the Lactobacillus family, show worsening of inflammations, whereas the treatment with postbiotics obtained through the Kalibiome technology shows maximum efficacy, tolerability, and a total lack of side effects. From a technological standpoint, Kalibiome postbiotics are obtained through a patented biofermentation process, implying 2 steps of fermentation rather than 1, with the second step carried out in a minimal culture medium through specific conditions which vary depending on the postbiotics output required. The knowledge of bacteria behavior and response during fermentation, along with the knowledge of the postbiotics' nature and selectivity of action, allows to design diversified groups of postbiotics with diversified activities. Kalibiome Technology was awarded at the IFSCC 2019, an extremely prestigious accomplishment, keeping in mind the strict parameters imposed by the judging scientific board of this organization. Following a table with an overview of the main advantages that Kalibiome postbiotics bring to the table compared to other microcategories of actives for microbiome care. In comparison to the prebiotics, which are the food of bacteria, Kalibiome postbiotics show higher specificity of action given by the presence of selected actives with specific function in their composition. Prebiotics instead are food for bacteria with no specificity or specific targeting. In comparison to the probiotics, which are living bacteria, we have 3 different types of added values. First, technological advantages, since Kalibiome postbiotics do not contain living cells. As a result of this difference, there is no theoretical or practical possibility of contamination or induced bacteria inactivation triggered by cosmetics preservatives. Another consistent technological benefit is linked to the storage condition. Given their chemical nature, Kalibiome postbiotics do not require refrigeration. In a second place, we have safety advantages, since Kalibiome postbiotics do not contain living bacteria, bacterial fragments, bacterial wall, or toxins. In the wake of the previously mentioned study, Kalibiome postbiotics show maximum safety and tolerability even in presence of acute inflammations. The probiotics instead, even commensal ones, in acute inflammation scenarios, could worsen the tissue's damages. The third advantage is functional. The probiotics, in order to give a beneficial action, need to crosstalk properly with the skin. Such crosstalking is quite facilitated in individuals with normal conditions, but it gets highly critical in individuals with uneducated skin. Now, one of the main examples of uneducated skin corresponds to the atopic skin. Individuals with atopic skin have unpredictable reactions when they get in contact with bacteria, even commensal ones. Kalibiome postbiotics, given their total lack of bacteria and their related structural components, overcome the need of cross-talking with the skin, as they are indeed ready to use actives whose benefits are observed regardless of the education of the skin. Other actives used in the market are the paraprobiotics, which are heat-inactivated bacteria or tyndallized bacteria. Their main benefit in comparison to the probiotics is linked to their higher stability in formula. When comparing them to Kalibiome postbiotics, they still show the same criticalities of the probiotics in relationship to the cross-talking mechanisms with atopic skin and uneducated skin, as well as safety matters in individuals with acute inflammations and uneducated skin. The last category are the lysates and ferment filtrates. These products are obtained through a standard traditional fermentation carried out with a single step where the bacterial output can be filtered or not. Compared to these actives, Kalibiome postbiotics show the following advantages. First, a higher content of reproducible and specific actives given the different criteria of the fermentation conditions. Second, a higher efficiency of action given the postbiotics' low percentage of use required in formula that starts from 0.1%. And three, optimized organoleptic features since Kalibiome postbiotics come in lyophilized powder form.